Imagine for a second, right, that you want to travel to Mars.
Okay.
But instead of letting you design a modern rocket from scratch, the rules say you have to start with an old, rusty bicycle.
Good luck with that.
Right. You are told to somehow tinker with it, like bending the metal frame, duct taping some thrusters to the spokes, adjusting the handlebars until it can somehow fly through the vacuum of space.
That sounds completely ridiculous.
It does. But for decades, that is essentially how scientists have had to approach biotechnology.
Yeah, exactly.
We took naturally existing proteins, the ones evolution provided over millions of years, and we just tinkered with them, trying to force them to solve modern, distinctly human problems.
And, I mean, it was an incredibly constrained way to do science. Natural proteins are beautiful, but they evolved to solve ancient biological problems, you know.
Right, like what?
Like they evolved to turn sunlight into sugar or to help a cell copy its DNA. But humanity faces very modern crises today.
We're talking Alzheimer's, climate change.
Exactly. We are dealing with Alzheimer's disease, with microplastic pollution choking our oceans, with a rapidly warming climate. And frankly, we just do not have hundreds of millions of years to sit around and wait for evolution to catch up.
No, we don't. We need molecular tools to fix these issues right now, which is why today's deep dive is just so thrilling.
It really is.
We're looking at a stack of recent research, and it's all anchored by this massive 2026 review article from David Baker's group at the University of Washington's Institute for Protein Design.
Published in Nature, yeah.
Right, in Nature. And the core takeaway for you listening is that the rules have completely changed. We are no longer trying to make bicycles fly.
No, we are typing spaceship into a computer.
Yay.
And it is printing out the exact atomically perfect parts we need.
Welcome to the absolute bleeding edge of de novo protein design.
De novo meaning literally from new.
From new. So our mission for you on this deck dive is to understand exactly how we made this leap. Like, how did we go from slow evolutionary tinkering to using generative AI to design bespoke molecular machines over a weekend?
It's a profound paradigm shift. We are talking about designing entirely new proteins from scratch proteins that have never existed anywhere in nature.
Which is wild.
Right. They're perfectly customized for the exact disease or environmental problem we want to target.
So to really appreciate the shiny new toys, we have to start by looking at the toolkit. How did the underlying science change so radically? Let's go back to the 90s and 2000s.
Yeah, so in that era, the entire field of structural biology was governed by something called Amundsen's hypothesis.
Amundsen's hypothesis.
Yes, it's a foundational rule in biochemistry. It basically states that the folded 3D shape of a protein is at its global free energy minimum.
Okay, let's unpack this. Global free energy minimum. That sounds very dense.
It does. But to visualize this, imagine standing at the top of a highly rugged, bumpy mountain, and you just drop a ball.
Okay, dropping a ball.
That ball is going to naturally roll down the slopes, bounce off ridges, and eventually settle in the absolute deepest valley at the bottom, right?
Makes sense. Gravity does its thing.
Exactly. For a protein, that deepest valley is its most stable, relaxed physical state. Because a protein is just a long string of amino acids, and it naturally folds up into the shape that requires the least amount of energy to maintain.
Oh, I see. So it just wants to relax.
Precisely. So early software, like a famous program called Rosetta, relied entirely on pure brute force physics to find that valley.
Brute force physics, meaning what exactly?
Meaning the computer had to mathematically calculate the physical forces between every single atom in a sequence. We're talking van der Waals forces, hydrogen bonds, electrostatic repulsion.
Wait, every single atom?
Every single one. It was computationally grueling. They were looking for the exact sequence of amino acids that would naturally roll down the hill into a specific stable shape they wanted.
It sounds like trying to build a massive jigsaw puzzle. But instead of just looking at the picture on the box, you have to calculate the exact friction, weight, and density of every single cardboard piece before you even try to connect them.
That is a great way to put it. It required evaluating billions of physical calculations just to see if a small protein would fold correctly.
But it did work eventually, right? The review highlights a protein called TOP7.
Right, TOP7. This was a monumental proof of concept.
Yeah, the very first entirely newly folded protein made this way. A completely synthetic shape that did not exist in nature, designed with atomic accuracy.
But while it proved the concept, doing the math atom by atom was just painfully slow. You could spend months of supercomputer time just trying to design one tiny, stable fold.
Which brings us to the big pivot. Around 2020, the field experienced a seismic shift.
A massive one. We moved away from trying to calculate pure physics, and we just handed the problem over to machine learning and AI.
Enter AlphaFold and RosettaFold.
Exactly. These deep learning networks were trained on the Protein Data Bank, or the PDB.
And the PDB is basically the encyclopedia of life, right? Like, it's a massive database containing 60 years worth of physically validated known protein structures that scientists have painstakingly mapped out.
That's exactly what it is. And these AI models looked at that vast database and learned the underlying hidden rules of how a sequence of amino acids translates into a 3D structure.
So they successfully mapped sequence space directly to structure space.
Yes. And that paved the way for the star of today's deep dive, which is generative AI. Specifically, a tool mentioned extensively in the sources called RFdiffusion.
RFdiffusion. Now, if you're listening, you've probably used an AI image generator like Midjourney or Daily, right? You type in a text prompt and the screen starts with a square of random static, just pure visual noise.
Right.
Then the AI slowly refines and denoises that static until, a picture of a cat riding a skateboard appears.
Yep.
RFdiffusion does the exact same thing, but for 3D molecular structures. It starts with a random 3D cloud of amino acids and progressively denoises it into a highly specific functional protein backbone.
And then a second AI tool comes in. It's called ProteinMPNN. It paints the exact amino acid sequence onto that backbone to make it a physical reality.
Like a 1-2 punch.
It really is. And it's incredibly intuitive once you see it in action. You can literally prompt RFdiffusion. You can say, I need a protein that perfectly binds to this very specific microscopic groove on a virus.
And it just makes it.
Yeah, the AI denoises a structure that physically fits that exact groove.
Okay, wait, hold on. I have to stop you there.
Sure.
I love the dolly analogy, but there's a huge gap here. If Midjourney messes up a cat's paw and gives it 6 toes, who cares? It's just pixels on a screen.
Fair point.
But a protein has to physically exist. It has to fold in the real world, survive in the hot, boiling, chaotic soup of the human bloodstream, and not just instantly fall apart.
Right.
So if this AI is just hallucinating shapes from static, how does a computer know about real-world physics? How does it know the protein will actually survive?
And that is the absolute genius of training the AI on the Protein Data Bank. The AI is not just creating a pretty picture. It is relying on 60 years of actual proven physical reality.
Oh, because it only knows what actually works.
Exactly. The older physics-based models we talked about calculated energy. But the new AI models calculate the probability of a structure existing based on the vast data set of everything nature has ever successfully built.
That makes so much sense.
It captures the intrinsic physical coupling between folding and function, because the AI only knows how to build things that look and act like the stable physical proteins it was trained on. It doesn't hallucinate impossible physics, because impossible physics weren't in its training data.
Okay, let me make sure I'm wrapping my head around this shift in a quick recap for you listening. We spent decades painfully calculating physical forces, atom by atom, friction by friction, to build new proteins.
Decades, yes.
And now we're using AI diffusion models trained on everything nature has ever built to just instantly hallucinate mathematically perfect, stable protein structures in a matter of days.
Basically, yeah.
We have essentially built the ultimate atomic 3D printer.
We have. And naturally, once you have an atomic 3D printer, the very first thing you do is start printing the basic physical infrastructure. The scaffolds, the containers, the building blocks.
The nanoscale Legos?
The nanoscale Legos, exactly. The sources detail how scientists are designing customized protein pores. Specifically, transmembrane beta barrels.
Okay, let's not lose anyone with the terminology. Transmembrane means it sits across the cell membrane. But what is a beta barrel?
Think of the cell membrane like a thick, impenetrable fortress wall protecting the cell. A beta barrel is essentially a microscopic hollow straw made of interlocking sheets of protein that you can insert right through that fortress wall.
So it creates a custom tunnel.
Yes.
But why do we need custom microscopic tunnels? Like, what's the application?
One massive real-world application is single-molecule DNA sequencing. Imagine you have a long strand of DNA, and you want to read its genetic code. If you design a custom beta barrel pore, you can literally pull that single strand of DNA through the tiny hollow tube.
And read it as it goes through.
Exactly. As it passes through, you measure the electrical disruptions, which allows you to read the genetic code in real time. We used to have to rely on whatever natural pores we could scavenge from bacteria, but now We can dictate the exact width, shape, and electrical lining of the tube.
That is incredible. We're just printing custom tunnels.
Yeah.
Now, the paper also mentions macrocyclic peptides. What are those doing?
So macrocyclic basically means a big circle. These are small, highly stable loops of amino acids. And their tiny size, combined with that tightly locked circular design, allows them to easily slip right through those cell membranes.
Meaning they can get inside the fortress.
Exactly. This makes them incredibly valuable as orally bioavailable drugs. You could take them as a pill, and they can sneak into spaces inside our cells that larger, traditional protein drugs simply cannot reach.
Incredible. But where it gets truly wild for me is when we look at higher order assemblies. Scientists are blueprinting proteins that act like interlocking Lego bricks to self-assemble into complex geometric shapes.
The assemblies are fascinating.
We're talking tetrahedrons, octahedrons, and icosahedrons. Now, an icosahedron is a 20-sided shape, like the 20-sided die you'd use in Dungeons and Dragons.
Right.
And what blew my mind is that this isn't just theoretical whiteboard math anymore. The review points out that this is already in human arms.
Yes, the SkyY-Covione COVID-19 vaccine.
Yeah. It uses a design icosahedral nanoparticle as its base. It is literally the first completely de novo designed, clinically approved medicine in human history.
It is a massive milestone. I mean, they built a microscopic 20-sided ball entirely from scratch, stuck the COVID virus spikes on the outside of it, and used that incredibly stable synthetic cage to safely train the human immune system.
That's so elegant. And the design principles are getting even smarter, right?
They are. What's fascinating here is how the designers are now pushing past perfect symmetry. For a long time, we could only build perfectly symmetrical, rigid shapes.
Because that's what the math allowed.
Exactly. But the researchers realized that if they intentionally introduce something called geometric frustration, they can build even more complex things.
Geometric frustration. That sounds like me trying to assemble IKEA furniture.
It's somewhat similar. Imagine you are trying to build a perfectly flat patio floor out of bricks, but all the bricks you have are slightly wedged. They are just a tiny bit thicker on one end than the other.
Okay, so they're uneven.
Right. If you keep laying them down, the floor won't stay flat. The intentional asymmetry, the frustration, forces the overall structure to curve upwards, eventually forming a massive dome.
Oh.
So by breaking perfect symmetry and designing subunits that don't fit together absolutely perfectly, scientists can force these proteins to curve and assemble into massive complex structures up to 100 nanometers wide.
So just taking a step back here, we've basically built the perfect microscopic delivery trucks.
Basically, yes.
We can print tiny geometric cages, hoops, and custom tunnels that assemble themselves on command. But here's the catch. A perfect delivery truck is entirely useless if the driver is blindfolded.
Exactly.
The truck needs a GPS. It needs to know exactly where to deliver its payload in the body. So how do we give these nanomachines a targeting system?
That is the crucial next step. A nanomaterial needs to know exactly what to bind to. And this is where RF diffusion truly shines. We mentioned earlier that you can prompt the AI.
Right.
You can give RF diffusion the precise 3D coordinates of a target, say a specific patch on the spike protein of a virus or the surface of a cancer cell. And the AI will generate a custom protein that perfectly hugs that exact microscopic landscape. They call these design proteins binders.
Binders. It's like asking the AI to design custom molecular Velcro. You just scan the thing you want to grab and the computer prints the exact corresponding hook.
Exactly.
And what's amazing is that it works on targets we used to think were completely impossible to grab. The paper talks about intrinsically disordered regions, or IDRs.
Yes, IDRs have been a massive headache in medicine. For a long time, they were considered completely undruggable.
Because they don't have a shape. Think of a normal protein like a rigid lock. You can design a key for a rigid lock, but an IDR is like a wet, floppy noodle.
Right, they're unstructured.
So if a protein doesn't have a rigid structure, how on earth do you build a drug to stick to it? The answer, according to the sources, is a concept called induced fit.
Induced fit is a brilliant structural workaround. Because the IDR is a floppy, flexible noodle, the design binder acts almost like a rigid mold.
A mold, okay.
When the floppy target encounters the binder physically forces the IDR to conform and snap into a specific locked shape.
So it's like, imagine having a lock, but the key is made of melted wax. When you shove the wax into the lock forces the wax to take the correct shape.
That's a perfect visualization. And this is incredibly important because many highly destructive disease-causing proteins feature these floppy, disordered regions.
Like Alzheimer's, right?
Yes. A primary example would be the amyloid and tau proteins, which are heavily implicated in Alzheimer's disease.
Oh.
We can now design traps that force those floppy, toxic proteins into a rigid structure, neutralizing them before they can form plaques in the brain.
The real-world examples of this custom Velcro are mind-blowing. They are using these binders to neutralize snake venom and the lethal toxins from C. diff bacteria.
Which is huge.
Think about the implications for snake bites in the developing world. Traditionally, anti-venom requires a strict cold chain, right? It has to be kept refrigerated from the lab all the way to the clinic, which is incredibly difficult and expensive in remote areas.
It's a huge logistical barrier.
But these de novo proteins, they are hyperstable. You can design a synthetic antitoxin that can sit in a hot backpack in the jungle for months, and it will still perfectly bind and neutralize a lethal snake venom on contact.
And we're going beyond just passively blocking toxins. We are actively directing cellular traffic. Scientists are engineering what they call endotags.
Endotags, so tagging something inside the cell.
Exactly. These are custom binders engineered to grab onto a disease-causing protein, like a rogue cancer receptor, and physically drag it across the cell to the lysosomes.
And lysosomes are like the trash cans, right?
The lysosome is essentially the cell's internal garbage disposal or recycling center. So we are not just blocking the bad proteins, we are tagging them, grabbing them by the collar, and throwing them directly into the cellular trash can to be degraded.
That is so aggressive. I love it. They are also designing binders for small molecules, right? Like targeting the drug fentanyl or apixaban or hormones like cortisol.
Yes, the sensitivity is amazing.
Which means we could have incredibly sensitive real-time biosensors that can detect microscopic levels of a specific drug in a patient's bloodstream instantly.
The sensitivity we can achieve when we custom design the binding interface is truly unprecedented.
Okay, here's where it gets really interesting. I have to ask the big question. If we can just ask a computer to design a binder, a custom piece of Velcro for literally any virus, any venom, any drug, or any floppy Alzheimer's protein, does this mean the fundamental challenge of drug discovery is basically solved? Have we beaten the final boss of pharmacology?
That is a very bold statement.
I know, but still.
But according to the consensus in this review, the long-standing technical challenge of how to design a binder for a specific target is indeed largely solved.
Wow.
The barrier is no longer the computational ability to create the molecule. The new critical bottleneck is simply deciding what we should target.
Oh, because biology is messy.
Human biology is breathtakingly complex. We now have the smart missile. The challenge is making absolutely sure we have the correct GPS coordinates so we hit the exact mechanism causing the disease without triggering unintended side effects in healthy tissue.
Let me make sure I fully grasp the magnitude of this. The era of undruggable diseases is effectively over. We can print molecular traps that force floppy Alzheimer's proteins into submission. We can cure snake bites out of a hot backpack. And we can physically drag cancer proteins to the cell's garbage disposal.
That's the reality we're entering.
We have the targeting system down.
We do.
Yeah.
But binding to a target is fundamentally a static action. It's just two things sticking together. Biology, however, is dynamic.
Right. It moves.
The proteins in nature are not just structural beams or pieces of Velcro. They are machines. They cut, they build, they shift shape, they react. The true final frontier of de novo protein design is creating AI-generated proteins that actively do things.
So we are talking about custom enzymes and biological logic gates. Let's look at custom catalysts first. In chemistry, reactions often have high energy barriers, meaning it takes a lot of heat or energy to force two molecules to react. Enzymes act as catalysts. They dramatically lower that barrier so the reaction happens easily at body temperature. And the researchers are using AI tools, like one called Placer, to design custom enzymes.
Yes.
Now, the paper mentions things like retroaldolysis and metallohydrolase. Let's not lose anyone here. What exactly are those two?
Let's break that down. A metallohydrolase is an enzyme that uses a metal ion, like a tiny speck of zinc, as a pair of microscopic molecular scissors.
Okay, tiny scissors.
It uses that metal to snip apart water molecules and break specific chemical bonds. A retroaldolase breaks carbon bonds. What the AI is doing is looking at the exact chemical transition state.
The transition state.
The awkward high-energy phase a molecule goes through right before it breaks apart. The AI builds a protein scaffold that physically forces the molecule into that exact awkward position, snapping the bond.
So we are literally designing microscopic metal-powered scissors from scratch to accelerate specific chemical reactions. That is incredible. And they are moving from chemical reactions to actual cellular computing. The paper details a dynamic mechanism called AND logic for cancer targeting. This feels like a massive leap for drug safety.
It's revolutionary.
Because traditional cancer drugs, like chemotherapy, are essentially carpet bombs. They attack any rapidly dividing cell, which causes severe side effects like hair loss and immune suppression. With AND logic, scientists are designing a protein system that requires two independent inputs to activate.
It brings computer science logic gates into human biology.
I love this concept. Think of it as two-factor authentication for killing a cancer cell.
That's a great way to describe it.
The designed protein floats around your bloodstream in a completely locked, inactive state. If it bumps into a cell that only has an EGFR receptor, nothing happens.
Right.
If it bumps into a cell with only a HER2 receptor, nothing happens. It only unlocks, activates its toxic payload, and kills the cell if it detects both the EGFR receptor and the HER2 receptor simultaneously.
It requires both molecular keys to turn at the exact same time.
This means healthy cells, which might naturally have one or the other receptor, are left completely unharmed.
Exactly. And if we connect this to the bigger picture, this kind of dynamic, programmable engineering extends far beyond just human medicine.
Oh, for sure.
The review explicitly outlines how protein design is moving toward planetary sustainability. For example, they're designing proteins for biomineralization.
Biomineralization, like how an oyster builds a pearl or a seashell.
Exactly like that. In nature, proteins guide the formation of inorganic materials like shells. Now, scientists are designing completely synthetic proteins that guide the formation of calcite from calcium carbonate.
Wait, really?
Yes. They're literally designing molecular factories to capture CO2 gas out of the atmosphere, fix it, and turn it into solid chalk. It is a biological approach to carbon sequestration.
They are also working on artificial photosynthesis, right? Like designing custom chlorophyll pairs to capture solar energy much more efficiently than plants naturally do.
With the goal of generating renewable solar fuels, yes.
We are talking about using synthetic proteins to manufacture semiconductors and energy materials from the bottom up.
It is a profound shift in how we think about manufacturing. Instead of top-down industrial processes that require massive amounts of heat, pressure, and toxic chemicals, we could use programmable, designed proteins.
Just to grow materials?
To grow materials, pull carbon out of the air, and generate fuel at room temperature, just like nature does, but optimized entirely for human needs.
So, to synthesize where this takes us, the next 10 years are going to be defined by nanomachines. We aren't just building static cages anymore. We are building biological logic gates that use two-factor authentication to hunt down cancer cells. And we are deploying molecular factories that literally turn greenhouse gases into chalk to fight climate change.
Looking at the entire landscape we've covered today, the implications are staggering. A process of random mutation and trial and error that took natural evolution 500 million years has been compressed into a deep learning model that takes a few days on a server.
It's hard to even comprehend that speed.
We've mastered the physical folds, we've built the geometric cages, we've designed the custom Velcro binders, and we are now animating these structures into working dynamic machines.
But of course, whenever we talk about a leap forward this massive, we have to address the elephant in the room.
Sure.
When you have a technology this powerful, an AI where you can literally type in a prompt and get a custom biological machine, what about biosecurity? Could someone theoretically ask RFdiffusion to print a supertoxin?
It is a highly valid and vital question, and one the field takes very seriously. The authors of the Nature Review address this directly, and it's important to understand their stance.
What do they say?
The paper notes that nature has already produced an abundance of highly lethal toxins and viruses on its own. The authors argue that the potential for these computational tools to protect humanity by allowing us to rapidly generate custom vaccines, antidotes, and diagnostics in a matter of days far outweighs the hypothetical risks of generating new dangers.
Right, the defensive capabilities scale just as fast as the offensive ones.
Exactly. Furthermore, they emphasize that the scientific community is actively developing stringent biosecurity screening tools to monitor and prevent the synthesis of any malicious AI-generated DNA sequences at the manufacturing level.
So what does this all mean for you listening today? We started this deep dive talking about the absurdity of trying to turn a rusty bicycle into a spaceship.
Right.
We are no longer constrained by the spare parts evolution left lying around in the garage. We have built the ultimate 3D printer for the fundamental building blocks of life. Nature took hundreds of millions of years to blindly evolve the proteins that make up your eyes, your heart, your brain.
And we are now doing it on computers with intention over the weekend.
So I want to leave you with this provocative thought to chew on as you go about your day.
Let's hear it.
If you could design a custom nanomachine to exist right now in your own bloodstream, whether it's a scavenger to clear out microscopic plastic pollution, a biosensor to detect a specific genetic disease years before the first symptom shows, or even an enzyme to enhance a natural biological function, what exactly would you prompt the AI to build?